Hello, everyone. I welcome you at the YouTube channel of Advanced Multiple Incorporation. Advanced Multiple Incorporation actually a knowledge forensic and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with Intellectual Consortium of Drug Discovery and Technology Development Incorporation. So both of these organizations are located in Mississauga, Ontario, Canada. I'm going to share my knowledge, my experience, and my research under the title of CRISPR-Cas9 and innovative gene editing technology. CRISPR actually stands for clustered regularly interspace palindromic repeat. So this is a technique. This is a like technology or protocol that is used for editing of the genetic material in human being or human cell. For this purpose, I have collected the information from different data sources, like it may be Medline, PubMed, Scopus, Google Scholar, National Library of Medicine, US, MBase, Directory of Open Access Journal, Duat, ProQuest, CNKI, that is China-based database company, provided research, scientific information all over the world. Some of the information collected from Crossref and ResearchGate. Before moving forward, I would like to discuss a little bit about the introduction of the CRISPR-Cas9. Genome editing or the genomic engineering is used to insert, delete, or replace the DNA in a particular segment of the genome for the betterment of human health. Clustered regulatory interspace short palindromic repeat, that is, as we discussed before, CRISPR technique has increased the speed and accuracy of gene editing. DuPont, Millipore, Sigma, and Selectis own the CRISPR-Cas patent. Jennifer is credited for the co-inventor of CRISPR. She is the first scientist who proposed microbial immunity mechanism that can be harnessed for the programmed gene editing. The first experiment was conducted by the using CRISPR to edit human embryo in 2015. CRISPR is now used for the detection and editing of the specific DNA segment. So, gene editing or the CRISPR-Cas9 can potentially be used for the treatment of different genetic disorder or genetic diseases. For this purpose, in this diagram, we are going to discuss a little bit about the use of CRISPR-Cas9 to find and replace the particular genetic material or the gene. We have divided the functionality or the function of CRISPR-Cas into the four step or four stages or four phases. In the first phase, a cell is transfected with an enzyme complex containing guide molecule and DNA cutting enzyme. These are the two very important components, two very important features and characteristic of CRISPR-Cas. First one is the guide molecule that take whole equipment or whole machinery at a particular desired spot. And the second one is the DNA cutting enzyme. This enzyme is taken from bacteria. The role of this enzyme to cut the particular part of the DNA. When it is cut down, then it is replaced after. So in second phase, a especially designed synthetic guide molecule find the target DNA strand. At this third phase, an enzyme cut off the target DNA strand. At the final stage, the amended DNA strand repair itself and start functioning like a regular gene. When we say that it start functioning, it means it start producing the messenger RNA. The messenger RNA that is called as transcription. The synthesis of messenger RNA, that is a transcription. When transcription completed, then these messenger RNA travel all the way down to the ribosome. In ribosome, these messenger RNA are translated and they produce particular desired proteins. So now we are going to discuss more about CRISPR-Cas system. For this purpose, we have divided the functionality of CRISPR-Cas into the four key components. At the first step, we identify and target the region of the genome to be modified. In second phase or in second step, the Cas9 enzyme unzip and cut the target DNA. The actual source of this bacterial enzyme is actually the Staphylococcus pyogenes. PAM sequence that is stand for the protospacer adjacent motif is part of target sequence DNA and is one of the factor that is required to define the cutting site. At the final step, guide RNA, a short fragment of the RNA bind to Cas9 that contain a recognized sequence matched with the targeted strand. With different guide RNA sequence, the Cas9 enzyme can be directed to recognize almost any kind of the sequence. The guide RNA lead Cas9 to desired location, bind the target sequence, and trigger Cas9 to cut both sides of the DNA. This double-stranded break provides an opportunity to edit the. In this diagram, I would like to talk a little bit about CRISPR-Cas mediated genome editing or the systematic CRISPR flow diagram. We most of the things we discussed already, like Cas9 enzyme, guide RNA, and the double-strand RNA, the PAM, target, cleavage, and DNA repairing. There is involvement of certain other component of CRISPR-Cas called as non-homologous end joining, nucleus lobe, protospacer adjacent motif, homology directed repair, and inserted and or deletion of nucleotide into the genomic DNA and include event less than 1 kb length. Okay, well, now, I would like to talk a little bit about genome engineering and CRISPR-Cas. The invention of CRISPR-Cas gene editing tool is great scientific revolution. There are almost 25,000 identified genes in the human embryo, and almost 300 linked to disease. Gene can potentially be edited in early embryonic stage. Researchers have used CRISPR in human embryo to repair a gene disorder, including heart defect, beta thalassemia, fatal blood disorder, and HIV. So, working on embryo may induce certain serious biological characteristics and change in behavioral pattern. So, if we talk about the cut and paste with the CRISPR-Cas, using a piece of guide RNA to locate a particular step in DNA, CRISPR then introduced the Cas protein, which cleave upon the target sequence and insert a modification to the genetic code. In this diagram, I would like to talk a little bit about nanoparticle delivery of CRISPR-Cas9 for the genome editing. There are the three main categories of nanoparticle used for the delivery purpose in CRISPR-Cas. The first one is Cas9 plasmid, second one is a Cas9 messenger RNA, and last one the Cas9 protein. CRISPR-Cas9 for in vivo gene therapy, promises and hurdles. This is the schematic illustration of CRISPR-Cas locus streptococcus pyogenes, site-specific DNA cleavage by nucleus Cas9. The resultant double-strand break or DSP are subsequently repaired either by non-homologous end joining NHEJ or by homologous directed repair upon the existence of the donor template. So these are the CRISPR-Cas9 manual and automated pipetting tools facilitate the process. These are the equipments manufactured and produced by different pharmaceutical companies. And in conclusion, the gene therapy revive as powerful treatment option for monogenic disorder. The emerging technology of biotechnology and molecular biology can potentially be used to unveil the fact and reveal the divine knowledge that will rationalize the ethical and philosophical interpretation of all these new inventions. The innovative gene editing technology will then be potentially help to achieve the desired clinical objective. At the last of this presentation, I would like to salutate and greet to my colleague, my team, and my coworker, Thierry Bajkar, Wolfgang, Daniel, Terry Mill, Robert Heyman, Arslan, Kunza, and Noreen. I acknowledge by this presentation to Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery Technology Development Incorporation. Both of these institutions are located in Mississauga, Ontario, Canada. Thank you very much for watching the video and staying with me for so long time. And see you soon. Take care. Bye.